All right. Can everyone hear me okay? Yeah? Okay. All right, so thanks so much for the introduction. Thanks so much to Ferhat and Sushmita for inviting me. It's a great pleasure, great honor to give a keynote here. I always love this meeting, particularly this year. I've just been blown away with the complexity of the field, right? Like where the field has gone over the last few years, especially in terms of integrating across modalities and single cell data and spatial transcriptomics. It's a really exciting time. I feel like I'm getting left behind a bit by the field because I am still stuck on a much more basic question in regulatory genomics, which is how does a transcription factor find its binding site in the first place, right? How do transcription factors recognize to bind particular locations on the genome and not others? And so while this is an old question, I still feel like we haven't solved it fully. So I'm sort of stuck on it for the moment. So let's go forward with this. So let's go back to real basics for a moment, and real sort of fundamental questions. Hang on a second. I think I need to, first of all, start a laser pointer and then do something else to advance the slides. Seth, sorry. Let me get a laser first. So that works. And then is that? Oh, okay. Thank you. Thanks, Seth. Okay, so again, back to basics. How do transcription factors find their binding sites in the genome? This question, of course, has a very fundamental but trivial answer, which is that DNA binding proteins, transcription factors, have DNA binding domains that form base-specific contacts with DNA. So, for example, here we have fork-head binding domain transcription factor, FOXA2. It has an alpha helix that fits neatly into this major groove and forms base-specific contacts, right? So every transcription factor has some molecular interactions that it makes with DNA, and we can characterize those interactions, of course, in high throughput. And for many, many transcription factors, this has been done. We can figure out which sequences these transcription factors like to bind to compared with other sequences. Of course, you can do this with a whole host of different in vitro and in cellular assays at the moment. You can do it with PBMs or selects or whatever it might be. And again, this has been done for many, many proteins. For most human transcription factors, we have some characterization of what they like to bind. So we can get all these binding sites, these sequences that these factors like to bind. We can line them all up. And of course, the old-fashioned way of doing things then was to make a simple position weight matrix model out of an alignment of sequences like this, right? So you might come up with a count matrix or something like that, some position-independent model. This is what we typically visualize, of course, right, as a motif. And then, of course, you can, if you want to predict where this transcription factor binds, you could, or you would have, years ago, have scanned this across the sequences that you're interested in to come up with some predictions on binding. And of course, as we know, this is almost useless, right? So let's take FOXA2 as an example, and this top sequence here is its favorite binding sequence, this TG-TTTAC 7-mer sequence. It likes to bind that more than anything else, any other 7-mer that you could come up with. But these short sequences are everywhere in the mammalian genome, right? So for example, this 7-mer sequence is present over 400,000 times in the mouse genome, about 500,000 times in the human genome. And when you actually look at where FOXA2 is binding in a relevant cell type, in liver, for example, you will see that only a very small proportion of those possible sites are bound. So for this particular sequence, this 7-mer sequence, only about 2% of those instances are bound in that given cell type. So there's this huge specificity problem. And of course, you can do this for any of the other sequences that I've listed here that FOXA2 might interact with. And the problem even gets worse, right? There's lots and lots of places where those sequences appear, and very, very few of those will be bound in a given cell type. So in general, for any given transcription factor, or for most vertebrate transcription factors, there are millions of possible sites on the genome where they could bind if you just look at in vitro binding data. And when you actually go and measure where they are binding in cells, only tens of thousands of those sites are bound. So this was famously called the futility theorem by Wyeth Wasserman, who was here yesterday, but unfortunately he left, so I can't make fun of the fact that this was like 20 years ago, where he coined this term futility theorem. His statement, maybe tongue-in-cheek, was that motifs are, it's it's basically futile to try and predict binding sites using these motifs because you're just going to be overwhelmed with false positives, right? There's a 2 order of magnitude false positive to true positive ratio. So why is this? So folks in the room here will of course know many of these reasons that this failed or that this was a bad approach to take. First of all, maybe these models, of course these models are too simple, right? So there might be more subtle sequence patterns in the flanks of these binding sites that factors will read in some way. There might be, for example, DNA shape parameters or DNA shape structural aspects of the DNA that will make some sites more favorable than others. There might be interdependencies between bases within a motif that might need to be modeled that will allow a transcription factor to bind or not bind. Beyond just sequences, though, beyond just having more complex sequence representations, there's of course also stuff we can just put under the broad heading of chromatin, right, of cell-specific chromatin environment. So for example, a lot of transcription factors will bind primarily in accessible regions of the genome, regions that are relatively nucleosome-free. A lot of other transcription factors, or a lot of transcription factors in general, will also have some cooperativity with other transcription factors or with other proteins, such that they might more likely bind to motif instances that are in some particular configuration with motifs from other cofactors. So all of these effects, of course, are cell-specific, and so that might explain why you see certain sites being bound in some cell types and not in other cell types. So while there's several recognized reasons that transcription factors will bind some of their motif instances and not others, we're finally getting to the point where we have models that can start integrating or at least handling some of these scenarios. And of course, most popularly in the field at the moment are neural network models that can both give you more complex sequence representations and allow you to integrate information beyond the sequence, right? More chromatin features. So these are good models for this problem. And I know most people here are familiar with neural networks, but just to re-emphasize why we think that they work well for characterizing transcription factor binding sites and other regulatory features of the genome. Convolutional neural networks in particular, which are quite popular in the space, we think of them as having the ability to recognize simple patterns within a data set and then combine those simple patterns into more complex representations of whatever you're trying to train them on. So in image analysis, for example, convolutional neural networks will train and will learn to recognize, first of all, simple geometric patterns like edges or lines or patterns of brightness or darkness or whatever it might be. And then in subsequent layers of the neural network, those patterns, those simple geometric patterns can be combined in different ways to give more and more complex recognition or more complex ability to recognize a particular pattern. So of course, this is what we think of when we think of applying convolutional neural networks to sequence problems. We hope that the same type of process might be happening. Now, of course, it's not looking at edges or anything like that within DNA sequences. We're often just thinking of the DNA sequence as a one-dimensional, like 4-color image, where the types of patterns that the convolutional neural network recognizes are more fundamental, simple motif patterns or motif-like patterns within the sequences. And they hopefully are then combined in different ways to give a more complex representation overall of whatever you're trying to train these neural networks to do. Right? So for transcription factor binding, we're hoping that these networks learn some fundamental patterns that the transcription factor likes to bind, or that are in the vicinity of binding sites, and that they are combined in such a way that it will give you an accurate prediction of binding. So these have been around for almost 10 years now, of course, and applied in this space, and they work quite well, right? They work much better than just motifs. If we put it in terms of AUPRCs, for example, and if I have an example here of FOXA1, looking at where it binds in HepG2 cells, the motif alone is almost useless, as I said, right? The AUPRC is only 0.01, compared with the neural network, where you get a much higher performance. Now, it's not perfect still, right? And this is one of my points today. I mean, if you even look at after correctly predicting half of the sites, right? Once your recall is 50%, you're basically making about 4 times more false positives than true positives, right? So there's still quite a false positive rate within these models. They're not perfect, but they're much better than what we had before, and they're now starting to be useful, right? And of course, we saw that in this conference quite a lot in different applications. We saw, and we will see, that you can train neural networks to predict transcription factor binding, and you can use those models to then perhaps interpret how genomic variants are affecting transcription factor binding or affecting other regulatory patterns. A nice feature of these models is that you're not just limited to DNA sequence. You can also integrate chromatin features, other measurements of a cellular environment, such as histone marks or accessibility or whatever you might have. And they can be used alongside sequence as just another feature space. There's lots of evidence where you can, or lots of examples of models where you can train it on sequence plus something from the concurrent chromatin environment. So for example, a TAC-seq. And you can then predict transcription factor binding on held out chromosomes in that cell, but by swapping out the chromatin signals here for the chromatin signal that appears in another cell type, you can make the neural network predict transcription factor binding in another cell type. And this works somewhat well. This works surprisingly well. This was the whole point of the ENCODE DREAM challenge at this conference about 5 years ago, I think, is when there was a challenge project within the DREAM side of this conference that was set up to try and predict binding across cell types. This was also, it's quite nicely illustrated by the max attack method, which was mentioned yesterday in a talk as well, right? So there's good methods for doing this. So what I want to draw a distinction on, though, is in this application, when you're predicting transcription factor binding across cell types, you're usually using some form of chromatin information that already encodes where the transcription factor is binding in some way, right? So you're sort of imputing transcription factor binding from something else that you've measured in the same chromatin environment. So we know that transcription factors are coincident with chromatin accessibility. By that I mean, where you see the transcription factor binding, for most transcription factors, you'll also see accessibility. So if you have the accessibility, you can impute the binding, right? What we're missing here in these various sort of applications is any real understanding of how that binding happened in the first place, right? So to illustrate what I mean by that, let's start thinking about things more temporally. Let's start thinking about a transcription factor that's becoming active into a given chromatin environment, and it has to find its binding sites, right? So now we can start looking at the same types of things I said before, accessibility and cooperativity. Maybe when a transcription factor becomes active in a given cell type, now you have a before and an after picture. And maybe in the before picture, there's already sites that are open that it can therefore bind. There's some sites that are closed that it can't bind. There might be some sites where there's already binding of some other cooperating transcription factors that it can latch onto and bind those regions, right? But we also know that for many transcription factors, there's also a cohort of sites that look like this, that are relatively inaccessible beforehand, but yes, the transcription factor can still bind them, right? And if we're only looking at the after picture here, if we're only looking at concurrently measured chromatin tracks, like a TAC-seq from the same cell type that you're interested in for transcription factor binding, you have no way of telling the difference between these sites and these sites, right? And you have no real mechanistic understanding of why the transcription factor is binding to these sites, whether it has like more ability to break into particular regions of chromatin, or whether it's dependent on the chromatin environment. So why this is important is if we care about trying to prospectively predict what would happen if you perturb a biological system, if you introduced a transcription factor into a given cell type, if you expose some cells to a particular hormone that results in transcription factor activation. We have a very limited ability to do that with our current data sets because we have no real understanding for most transcription factors about how much they'll just go along with whatever is accessible already, or to what degree they'll be able to break into new sites in a pioneering-like fashion. Okay? So what I'm really focused on today is I'm not going to solve that overall challenge, but I really do want to dig more into this question about how newly activated transcription factors integrate into an established chromatin environment. How sensitive are transcription factors to regions that are already open, or whether they can override chromatin state to some degree. So we're going to focus for most of the talk on a particular family of transcription factors that I've already mentioned, these FOX transcription factors. So they all have fork head binding domains. Reason we're focused on these is that one side of this family, one subfamily within the fork head transcription factors, are famous as pioneer transcription factors. They were some of the first pioneer transcription factors to be characterized by Ken Zaret. So by pioneer transcription factors, where this term came from was Ken Zaret and others recognized that some FOXA transcription factors have the ability to bind to nucleosomal DNA and then to establish competency for other transcription factors to bind. So in other words, they're able to open up those regions of chromatin to allow other transcription factors to bind, right? So this is sort of from their, a cartoon from their papers where the FOX transcription factor binds and then opens up a region. The way they're able to do this has been long thought to be within the, like core to the fork head domain itself. The fork head transcription factor binding domain, like I showed you before, it's this simple sort of helix loop helix structure that fits into the major groove, as I said. This structure is very, very similar to the structure of the linker histone. And there's this thought that's been going back decades whereby maybe the FOX transcription factors are substituting in for the linker histone and then destabilizing chromatin. And there is actually some evidence really recently from cryo-EM structures that this might be happening. However, we also know that this ability that FOX transcription factors have to pioneer new sites is not universal. If we look at where fork head transcription factors bind in different cell types, if we look at where a given FOXA transcription factor binds in different cell types, we'll often see it binding to very, very different locations. This has been done by many, many people. This is one example of a Venn diagram from a paper that did this in 3 different cell lines for FOXA1. It's looking at where FOXA1 is binding, and there's very little overlap between where it binds in these different cell types. So this ability to bind to nucleosomes and break open into those nucleosomes, it's not able to do that just universally. It's not able to do that at the same sites in all cell types, right? Or else there'd be a perfect overlap in these veins, right? Okay, so we wanted to dig into this a bit further, and we wanted to see how this ability or this non-ability to interact with relatively inaccessible chromatin might vary across different fork head domain transcription factors. Because the FOXA transcription factor family has been well characterized, but very little is known about whether the other fork head transcription factors do the same thing, even though they have very, very similar DNA binding domains. So they all, in other words, have DNA binding domains that look like the linker histone. So if it's really the linker histone similarity, then they all should be pioneers, but that actually hasn't been characterized. So there's about 45 or 46 paralogs of FOX transcription factors in mammalian genomes. We're going to look at a selection of them here, just a selection across this phylogeny of different fork head transcription factors. We're going to specifically look at FOXA1, FOXC1, FOXG1, FOXL2, and FOXP3. These are all important developmental regulators. They're all involved in specifying very different lineages during development, different cell types, different tissue types. Importantly, according to the in vitro data at least, they all bind very, very similar motifs, right? So if we look at what's been characterized for these fork head domains in terms of PBM characterization or however these have been measured in vitro, they all bind very similar TG, TTT, AC patterns. So we want to see, again, if they have the ability to pioneer into new particular sites when you introduce these factors into a given chromatin environment. So basically, this is our question here. Will these various FOX transcription factors that have identical, very similar in vitro binding preferences, will they all bind to the same sites if we introduce them into a given chromatin environment? So it's quite a simple setup here. We're just going to induce them in mouse embryonic stem cells where they are not naturally expressed, because we want to introduce them into a setting where they are not already present and see what they do with the chromatin that they're faced with, right? We're going to measure the binding using ChIP-EXO. We're going to look at the outcome of that binding with RNA-seq and ATAC-seq. ChIP-EXO, for those of you who might know, is just a high-resolution version of ChIP-seq. There's an additional step in here where there's an exonuclease used to digest the DNA in a single-stranded fashion back to where the protein is actually binding, or where it's cross-linked at least to the DNA. So you get your sequencing reads start right at the position where the protein is protecting from the exonuclease. So the effect of this is you have much higher resolution data. So we did this for these 5 FOX transcription factors, and at some of the sites, we see that they do in fact bind similarly. So at some sites, all of these FOX factors are binding to those sites in a similar fashion. But at many, many more sites, it looks more like this, where some of the FOX factors will bind to sites, say, for example, here, where FOXG1 and FOXP3 are binding, but the other FOX factors won't. Likewise, here's one where FOXA1 is binding and the others aren't. When you look across the whole genome and cluster out the different patterns that you see for all possible sites that are bound, you get something like this, where, again, you can see that there's quite a range of binding affinities to different sites according to these different FOX paralogs. Again, even though they've been expressed in identical chromatin environments. So, for example, there's this large cluster of sites here that are preferred by FOXA1. There's another cluster down here that's preferred by FOXG1 and P3. There's yet another cluster down here that's preferred by FOXL2. So why, again, if we're introducing them into the same cell type, why is there this diversity? Why are there these differences? The first thing is that there is some differences in how these transcription factors, in what pattern they like to bind in cells. And this is one thing that we have to be careful about. You know, when we've measured all these motifs in vitro, they're not necessarily binding the same thing within cells, right? So when we look at what the most popular motif is for these various sites, the FOXA1 sites, they show the motif that it should be binding in vitro. This is the one I showed you in vitro before, this TG-TTTAC pattern. But most of these other sets of sites have a related but distinct motif that's more like a repetitive version of this TG-TTT pattern. So you sort of see TG, TTT, G, TTT, G, TTT, this sort of tandem repeat of this simpler related pattern. But crucially, we don't see different motifs amongst these different binding patterns. So for example, the sites where FOXG1 and FOXP3 are binding and not L2 has the same motif present as the sites that are bound by L2 and not G1 and P3, right? So it's not all down to sequence. Sequence is definitely a component, but not fully. So this is with meme, and this is with the simple motif finders. You know, we got to make sure that we're not fooling ourselves here and that there's not some underlying similarity or underlying differences rather in the sequence patterns that are being bound. So of course we turn to neural networks. We trained a convolutional tower neural network to predict the ChIP-EXO read depth at all sites across the genome. So we're trying to predict the quantitative level of binding, proxied by ChIP-EXO read counts. And we're doing this with only sequence features to begin with here. So when we do that, and then we test, of course, on held out data, let me explain what I'm showing you here. On the y-axis of these graphs here is the true ChIP-EXO read count for all these sites, the log read count rather of these for these sites. And here's the predicted on the x-axis. So if this was working correctly, you'd see it spread along the diagonal, right? If you're getting good recapitulation of the ChIP-EXO read count, and you don't see that very well here, right? It's not doing a great job at really predicting the read count from sequence alone. And I can show you a bit more clearly if we sort of ask it to make more solid predictions on these clusters of sites. You can see that really it's not doing a very good job at recapitulating the ChIP-EXO when we've trained on sequence alone, right? So maybe if you squint, you can say that the FOXA1 network is given more preference to these sites that are bound by FOXA1 than the other sites. But look at something like FOXG1 here. It's basically doing terrible across the whole range of sites, it's not really doing a good job at distinguishing the fact that these sites are bound by G1 and the others aren't, right? So sequence alone isn't really doing the job. The one thing that is good about these sequence models is that you do have some interpretation of what they're learning. You do have some ability to do some feature attribution and to figure out which sequences at these peaks are driving a particular prediction. So just to give you an example of that, and this will become relevant later a little bit, here's a site that's bound by FOXA1. Here's just two different views of the chip exo data. This is like the 5' read counts that are really sort of stopping where the proteins are protecting the from exonuclease. This is a smoothed version of that. And this is a site that's both bound by FOXA1 and that's predicted by the FOXA1 neural network to be bound. And then we can ask the neural network what sequences within this region are important for driving that prediction. So if we zoom in on this exact same site here, you'll see that it's highlighting, I'll have to highlight these for you, it's highlighting particular motif instances that FOXA1 likes to bind, right? So everything sort of makes sense in this region, right? So this is a neat way of drilling into the sequence features that are important for predictions. You can do this at all sites and make heat maps out of the sequence attributions. And again, it's the same sort of story that I showed with just the raw predictions themselves. If we look at the sequence attributions, the FOXA1 neural network does distinguish these sequences that are actually bound by FOXA1 as being different from the others. It is recognizing some features at these sites that it's thinking as more important or more compatible with FOXA1 binding than other sites. But again, look at the FOXG1 pattern here. It basically thinks that all of these sites should be bound in a way, right? It's seeing sequence features that are compatible according to what it's learned about FOXG1 binding. Is finding these sequence features that are compatible with FOXG1 binding at all of these sites, even though only a subset of them are actually bound by FOXG1. Running this through, running these sequence attributions through something like TF-MODISCO doesn't really help to clarify anything from the standpoint of further understanding, because it's finding the same types of patterns that we found just from meme, right? It's just really finding the fact that FOXA1 really likes this TG-TTT-AC pattern, and the others all bind to similar repetitive patterns, right? Okay. All right, so that's from the sequence view. But remember, we've overexpressed all these FOX factors in exactly the same chromatin environment, these mouse ES cells, and there's quite a lot known and quite a lot profiled in mouse ES cells. So there's a lot known about other chromatin features. There's a lot of work from many, many labs that has collected basically everything you'd ever want to look at in mouse ES cells. So the question is, are these FOX factors interacting with chromatin features in different ways, right? And we can ask that question because we know the before state of the chromatin from all those other studies. So we can do something really simple to begin with. We can look at where these FOX factors are binding and look at the overlap between those binding sites and stuff that was happening in mouse ES cells before these FOX factors were expressed. So this is what we're calling the pre-existing chromatin environment, right? So before these FOX factors were expressed, the sites where they're binding have various degrees of overlap with pre-existing chromatin features. For example, FOXP3 binding sites, over half of those sites were already either accessible or marked by relatively active histone modifications in the pre-existing mouse ES cells before FOXP3 was expressed. On the other hand, FOXL2, most of its sites were not already accessible, or were not already marked with active histone modifications, or with repressive histone modifications before FOXL2 was expressed. You can sort of look at this in another way through chromHMM, where of course we'll make chromatin state patterns. And again, the same basic message is there. Most of FOXP3 sites, most of FOXG1 sites are already in regions that were marked as enhancers or promoters in ES cells. A much larger proportion of FOXL2 and FOXC1 sites are in regions that are basically called by chromHMM as quiescent, so basically devoid of most histone modifications or accessibility. Okay, so that's sort of, again, just a basic overlap, but can we drive a bit more understanding of what's going on with neural network methods? So again, we turn to the same type of convolutional tower architecture, and now instead of just using sequences, we're throwing in a lot of different chromatin tracks from the pre-existing chromatin environment. So accessibility, histone modifications, etc. But from the mouse ES cells, again, before binding. And we're trying to predict binding after expression, right? So does this help in explaining or in predicting where the transcription factor will bind? The quick answer is yes. You get a drop in mean squared error as a proxy of performance here. By incorporating chromatin features into the neural network, you get improvement, in other words, in the ability of these neural networks to predict the ChIP-XO read counts. And it's proportionally a higher improvement for some factors than others. So for FOXG1 and FOXP3 in particular, you get a proportionally higher improvement in performance. We can compare, the same scatter plots as before, looking at the predicted versus observed ChIP-XO read counts. And now you can see that for some of these factors, at least, you're starting to see that spread along the diagonal that would indicate it's actually doing something useful, right? So especially for FOXG1 and FOXP3, you're getting much tighter correlation with the real read counts at these various sites. Again, after incorporating chromatin features into the model. Here's the view from the actual clustered site. So this is what I showed you before, the sequence-only neural networks. And this is the same predictions at the same clusters of sites after integrating the pre-existing chromatin features. It's not perfect. I'm not going to claim that there's no improvement to be had here, but it's starting to at least discriminate these sites that are bound by FOXG1 and P3 from the others at this point, right? You can start seeing that it's improving performance on these sites to some degree. So just as we had looked at our sequence feature attributions, we can also look at our chromatin feature attributions. We can run the same types of feature attribution techniques on the chromatin features instead of sequences and ask what chromatin features the neural networks think are important at these different sites that are predicted by the models. We can run that again, just like I did with sequence, you can run it across the whole genome. And you start seeing some interesting patterns in here. So for example, you'll recall I showed you this cluster of sites that are preferred by FOXL2 and that are not bound by FOXG1 or FOXP3. And the sequence side of the model, as I'll show you in a second at a couple of sites, the sequence side of the model thinks that these sites should be bound by FOXG1 and FOXP3. But the chromatin side of the model sees that there's no attack-seek signal at this cluster of sites. And it uses that as a negative feature to predict FOXG1 and FOXP3. So even if there's sequence features present, the network has learned that that's not sufficient, that it needs accessibility as well. So this is true for attack-seek. This is also true for a mark like H3K4 monomethyl. I'm going to show you this briefly at individual sites here. So this is an individual site that's bound by FOXL2. Again, we have a couple of different views of the ChIP-XO up here. When you run the sequence attribution on this model, this is also a site that's predicted to be bound by the FOXL2 model. And so now we're going to ask what features are contributing to that prediction of binding. The sequence attributions highlight sequences that we've already know here are preferred by FOXL2. These TG-TTT patterns are related. There's no prior accessibility or K4 monomethyl signal at this particular site, right? You have very low read counts for both of those at this site. And what I'm trying to show here, maybe the visualization could be improved, but these are the chromatin feature attributions. And you can see that there's a slight, the 0 is right here in the middle. So basically the model doesn't care really one way or the other whether there's attack-seek or not at this site. These are very slightly negative attributions for the attack-seek and for the K4 monomethyl. If we look at the exact same site for FOXA1, FOXA1 does not bind to this site. There's no ChIP-XO signal. The FOXA1 network does not predict it to be bound at this site. But that's really because the FOXA1 network is not seeing any sequence features that it likes at this site, right? You see nothing in the sequence attributions that would predict FOXA1 binding. But a very different story comes when you look at FOXG1. Again, at the exact same site, FOXG1 does not bind this site either, but the FOXG1 neural network loves this location. It sees lots of sequences that it thinks should predict binding. But those positive features are being counterweighted by these highly negative features that are associated with the lack of ataxic or H3K4 monomethyl. So I really like the ability to drill into these individual sites and identify, for example, sites that you could predict would be bound if we looked at them in a site in a cell type where they were accessible. So for example, if we change the accessibility here, this negative feature will go away, and maybe you would see binding for FOXG1 in that case. There's real, like, you know, solid hypotheses you can generate. Not that we've tested them yet, at least, but you know, I like the ability of these models to go into that detail. So in summary, on this part at least, and this is a cartoon, this is not quantitative axes here, but this is the way I think about it, that these different FOX factors have different degrees of dependence on the pre-existing chromatin environment versus their innate sequence preferences. So we can think about putting these different factors along this spectrum where some of them, like FOXP3 and G1, are more dependent on the pre-existing chromatin environment. Some of them, like C1 and L2, are less dependent on the chromatin environment, but more driven by their sequences, right? So once the binding has happened, of course, then the real question about pioneering activity really comes from what happens next at those sites. So as I told you before, we've done the ATAC-seq on these different on these different factors as well. So here we're looking at sites that have already split out into, according to their status of whether they were accessible or not in mouse ES cells. And so the different degrees of separation here or overlap here are, of course, just what I told you already about FOXP3, for example, just primarily binding in accessible regions, and FOXL2 binding a lot more sites in relatively inaccessible regions. But what we're interested in here is what happens next, what happens after the binding. Is accessibility created? So I think it's going to be more clear if you look at this view here, where again, I've separated the previously accessible and the previously inaccessible sites. And then these colored lines are going to show the average ATAC-seq signal that's generated after binding of the factors. You can see the FOXA1 results in a huge increase of accessibility at its binding sites, which is expected because it's a known pioneer factor. FOXL2 and FOXC1 drive accessibility at their binding sites, but to less of a degree. And FOXG1 and FOXP3 generate no more accessibility than we would have seen from a control line where we didn't induce any factor and we just let them differentiate just randomly. So not only do we have like an axis of dependence on pre-existing chromatin, but I think there's also another set of axes to consider in terms of how much these various transcription factors have an ability to create accessibility at their binding sites, right? And I don't think we can really start classifying different transcription factors according to these different axes until we do a lot more experiments where we're inducing transcription factors and measuring the effect, measuring where they bind, measuring what their effect on accessibility is. We're not going to get this type of information from just characterizing binding in static cells and cell lines or in other tissue types where we're not making any perturbations. So we're having a hard time publishing this, by the way, because as soon as you get into this type of viewpoint, the question that comes up with even journals you wouldn't expect it to come up with is, where's your mechanism, right? Why is there a variation in the ability of different FOX factors to bind to inaccessible chromatin or to make that chromatin accessible? So it's been a bit of a headache over the last while trying to just sort of either make this story publishable or actually go and derive mechanisms. So we have actually started going down the line, which I never thought I'd say, but we have started going down the line of trying to mechanistically determine what parts of the proteins are responsible for pioneering activity or not. So for example, there is some evidence in the literature that there's a part of FOXA1, this part of the C terminus that might be important for binding to nucleosomes and for creating accessibility. And so we're gone down the old-fashioned route of doing chimera and truncations of the proteins and overexpressing each of these one at a time into mouse ES cells. And then we're going to maybe try to take this part of the protein and stick it onto another FOX transcription factor to make it a pioneer factor. So we'll see how that goes. I don't have huge amount of results yet, but one that I wanted to share that's really driving me nuts, I can't really understand what this is telling me at the moment, is we've at least done this one to begin with, just the FOX DNA binding domain alone, right? And if we've taken away all these other parts of the protein that are hypothesized to be involved in its pioneering activity, and we just have the raw DNA binding domain, I thought that we'd just get binding in pre-existing accessible regions. And we found the exact opposite. So first of all, we got really beautiful ChIP-EXO data out of this experiment. It's binding primarily to its cognate motif, in fact, to a sort of extended cognate motif. And almost all of those sites are in regions that are inaccessible. And furthermore, we're just doing this, it's on the sequencer now, but I don't think these sites are becoming accessible. I think it's just binding to inaccessible regions and just staying there and not doing anything with those regions. So that's, again, this is a little confusing to think about for me at the moment. So the message here for this part of the talk is that we have these various paralogous transcription factors with very similar DNA binding domains and even similar in vitro binding preferences. And if we're just to look at where they bind in a static cell type, we would see them binding to different regions on the genome, and we would not see too much of a difference in what motifs they're binding to. And what we'd be completely missing is the fact that there's this huge difference in what the chromatin looked like before binding, right? So there's a big difference in, say, FOXP3 that primarily binds to accessible regions, and FOXL2 that primarily binds to inaccessible regions and makes them a little accessible. So that's one message here, is that we think paralogous transcription factors can diversify their binding preferences by not just varying the sequence preferences, but by also varying how they interact with pre-existing chromatin environments. I know I'm running out of time-ish, right? But I will say that this is the second set of paralogous transcription factors that we've noticed this pattern in. In a different collaboration a few years ago, we were looking at Hox transcription factors. These are a famous, important family of transcription factors, of homeodomain transcription factors that are deeply conserved and that are involved in specifying positional identity of cells along the growing embryonic axis. So they are expressed along the embryo axis in the order in which they appear on the genome. There's 4 clusters of Hox factors in mammals, each with some subset of 13 paralogs. One of their jobs in specifying positional identity is within the motor neurons in your spinal cord. There's different, depending on which Hox factors are expressed, you will get motor neurons extending axons out to your upper limbs or to your body wall or to your lower limbs, right? So these are really important in telling a motor neuron what it should be doing. But these Hox transcription factors also have very, very similar in vitro binding preferences to one another. There's really just 2 forms of patterns. They either bind this sort of TAAT pattern or this TTTAT pattern, depending on how deep you are into the paralog copy number. With Esteban Mazzoni at NYU, a few years ago at this point, we had a project where we overexpressed these factors one at a time in progenitor motor neurons, and we could make those motor neurons become limb contacting motor neurons or body wall contacting motor neurons or lower limb contacting motor neurons, depending on which Hox factor we were overexpressing. And when we started looking at where these Hox factors were binding in the genome, again, just like with the Fox factors, we found that those Hox factors were binding to different sites on the genome, and that we couldn't fully explain these differences with motifs. So for example, there was this large set of sites that HOXC9 liked to bind that had an identical motif to where you had binding of both HOXC9 and HOXC10. Those 2 paralogs, HOXC9 and HOXC10, have very similar binding preferences, but that's the difference between getting motor neurons that are body wall versus lower limb contacting motor neurons. And that's really down to these sites that are only bound by HOXC9. And really the difference, again, just to shortcut this story a lot, is that these sites that HOXC9 likes to bind were primarily in relatively inaccessible chromatin regions in the progenitor motor neurons. So again, we think that HOXC9 had this greater ability to interact with pre-existing inaccessible regions, right? That it was maybe more of a pioneer factor, for want of a better descriptor. At the time, we thought that we were also dealing with sequence versus sequence plus chromatin neural networks in that story. And this is sort of a similar picture, although it's in a different format, but a similar picture to what I showed before, that the relative increase in performance when you integrate chromatin into your model is maybe an indicator of how much a transcription factor is dependent on pre-existing chromatin features. So for HOXC6 and HOXC10, which we think are quite dependent on the pre-existing chromatin environment, there was this larger increase in performance when we integrated chromatin into the model compared with HOXC9. So again, this was an example where we think that these HOX factors, these paralogs, are diversifying their binding sites based on interactions of chromatin as opposed to the interactions with sequence alone. So one very last point to make is that all of this is dynamic. So when you have transcription factors coming in and binding to regions of chromatin and opening up those regions of chromatin, that of course is going to affect the next wave of transcription factors that becomes activated. And those transcription factors, if they are sensitive to that chromatin environment, will then be dependent on the accessibility that's been created. One very last vignette here, very quickly discussed, is that we saw this in action for pro-neural transcription factors in a different project. Depending on which basic helix factor you express in mouse ES cells, if you express ASCL1 or NeuraG2, you get two different flavors of neuronal progenitors, some of which are really going to become spinal motor neurons or sensory neurons, versus serotonergic and noradrenergic neurons for NeuraG2. Again, with these two factors, we saw huge differences in binding. But for these factors, we do not think that they interact differently with chromatin. This difference in binding was really just driven by a very simple difference in the motif feature. Very, very subtle difference in the motifs and the E-boxes that these two factors bind leads to a large difference in binding specificity on the genome. But again, no real difference in how it interacts with previously accessible or inaccessible chromatin. But my point here is that these two pro-neural factors, they're making neurons of different flavors, and you need a whole host of other transcription factors to become active in those cells just to get those cells to become neurons in general. So both of these factors activate a whole host of other transcription factors that are just general neural transcription factors. And these factors, we saw a pattern where these secondary neuronal transcription factors were differentially sensitive to this chromatin environment that was just been set up by the pro-neural transcription factors. So again, you see differential binding of these downstream factors to different degrees based on how sensitive these downstream factors were to chromatin or not. All right, so with that, the basic messages I wanted to give are that paralogous transcription factors can diversify binding according to these different axes, not just sequence, but also ability to engage with relatively inaccessible chromatin. We think neural networks are a really good way of digging into these differential determinants. And yeah, I guess we also see that these factors have a spectrum of abilities to engage with inaccessible chromatin. Most people think of pioneer factors as this separate class of transcription factors that we can just catalog and say, these are the pioneers and everything else is not the pioneers. And I think biology doesn't deal with binary very well, right? So there's a much bigger spectrum of abilities for factors to be somewhat sensitive to chromatin or somewhat more independent to pre-existing chromatin. So with that, I want to thank everyone who is involved in the work here. This is my lab, and a lot of what I talked about was in collaboration with either Esteban Mazzoni's lab or Frank Pugh over in Cornell or Minoru Ko in Keio University. Thanks so much.
I'll ask all three then.
I have 6. So.
Okay. All right. Yeah, I have multiple questions, Shaun. This is great. So one comment, I think that's not going to help you with your review, but great science opens up more questions than answers, right? And I don't know if you can write it in your response document somehow, but that's why I have a lot of questions. One is about the pioneering activity of these transcription factors, and then whether they are pushing ES cells towards some different trajectories of differentiation, and whether you are doing it in an acute manner that doesn't yet happen, that these are still pretty much ES cells.
Yeah, so the FOX factors, we also measure downstream expression. Now we're measuring binding and accessibility at a really early stage, like 12 hours after expression of these factors, right? So there's not like a huge developmental cascade has happened yet. Look, none of these factors that we're expressing should be naturally expressed in mouse ES cells. And that's really one sticking point with reviewers. They want physiological relevance, right? No, but to us, that was the whole point. We wanted it expressed in an unnatural environment so we could see the relative dependence on chromatin. However, we do see some relevant groups of genes being expressed. Like we do see endodermal genes being expressed downstream of FOXA1. We see some more blood lineage, like T cell lineage for FOXP3, for example. So there is some hints. I wouldn't say we're making T cells with FOXP3.
It's quick enough, I think. The other maybe suggestion is that like it can also be about what is there in terms of like other transcription factors and the co-bond things. And I don't think that data is published, but Mitch Gutman from so he had this chip dip data in mouse ES cells where they have done these highly parallel chip-seq, not the cleanest, not like chip exo profiles, but they have over 100 transcription factors that are represented, all done in the mouse ES cells.
Oh, nice. Oh, I haven't seen that.
Yeah, I mean, like you could have that essentially baseline, or that can be addition to your predictors. To add all the transcription factors that you can have, because you're using mouse ES cells and that data is there. I think that can help you. Yeah,
this also came up. I mean, that is a great suggestion, and I will dig into Mitch's data. There is a lot of chip-seq data already from mouse ES transcription factors. I actually have a hidden slide in here where we looked at the overlap with other transcription factors. We don't see anything specific that's in addition to just the general association with the TAC-seq.
Okay.
Does that make sense? Like we don't see a specific indicator that it's only binding OCT4 sites or something like that. But it's still a valid point that it could be interacting with something specifically that we're not measuring yet. So I'll dig into that. Thank you.
Really great talk. Thank you. I'm a big fan of pioneer transcription factors. I used to study the obligate heterodimer of ASCL1 and NGN2. So I know for pioneer transcription factors, the mechanism of initiating the unwinding chromatin state can relate to the exact positioning on or around the nucleosome. So I was wondering, for this family of transcription factors, do they exhibit like a universal binding on and around the nucleosome? Is it at those specific boundaries? Have you been able to kind of overlay those signals with, I guess, like inferred as well as you can nucleosome positioning?
That's a great question. So the question is like, we have high resolution binding from our chip exo. Can we localize where on the nucleosome they might be binding? And we did look against as high resolution MNase-seq data as is out there from mouse ES cells, and we didn't see any specific positional interaction. It's not just at the dyad, for example, or it's not just at the flanks. But the caveat there is that nucleosome positioning is, even the highest resolution that's out there is still very fuzzy. It's not very well, it's either not very well measured in mouse ES cells, or the reality is that nucleosomes are not very well positioned in ES cells. So I don't think we can't answer that at the moment.
Thank you. A quick question, we don't have to go into it. Do they also form heterodimers or homodimers, or do they need to dimerize?
For foxes, no.
Okay.
Thank you. It's messy with the basic helixes, though. Thank you.
So I have many questions, but I'll only ask one. So I have a question about like the first slide that you showed, which was comparing the performance of a motif versus CNN-based predictor.
Yeah.
What did the CNN predictor actually see? Like, you had to train it. Did you give it chip-seq data and make it like predict, classify peak versus non-peak?
Yeah.
And did you give the motif predict? Like, how did you make the fair comparison? Or this
is maybe a bit of a straw man, right? But like I was trying to come up with a way of showing that the neural networks are doing better. So the CNN in that context was doing a within cell type prediction, right? We fed it chip-seq peaks. I think we were asking it to predict the chip-seq read count in that context, and we were predicting the performance I showed is on held out chromosomes, right? So even in the within cell type, it's not perfect, right? And that's typical for some factors. Some other factors you get much higher AUPRCs. Like CTCF is almost perfect, right? But that's its own case. The motif, we're just looking at the score that comes out of the position weight matrix scan, right?
It's like it's an unsupervised versus supervised comparison. So giving CNN a little bit more information.
Oh, absolutely.
But if you were to like look at like there's the peaks, because here you are, in a way, you're not calling peaks. I mean, that's another follow-up question. Like, exactly what are you giving? Are you giving the entire genome and making it distinguish, like making it figure out, right? Is it a zeros versus non-zero and also called peaks? Or are you actually giving it regions with peaks and without peaks? And so you're really reducing the space that the CNN is being given. versus the motif, and you're like giving the entire DNA sequence, right?
Yeah.
And you're trying to make it find motif
Yeah.
in there, and so that's like a much harder problem. But if you were to like train it on just were to find like de novo motif finding on just the peaks, how would that a simple motif based predictor work?
Well, that's the paradox with motif finding, right? You always get the motifs out there. They are significantly enriched in the peaks, but yet they're just not specific when you look across the whole genome, right? At least with the motif scanning. Now there's plenty of other things you could do that's not a CNN. K-mer SVMs or whatever do quite well in this problem as well, or even SVMs trained with the full scoring range of the motif itself. Like it captures some multiplicity in the scores. But yeah, so this is again.
And maybe the performance may improve just a tiny little bit. But yeah, I don't question. I'm sure like the CNN is definitely doing better. But it's like an unsupervised or supervised problem.
Yeah, that's true.
Like
You're right.
Life is harder for the motif-based predictor.
That's fair.
Yeah,
that's
waiting
fair.
for your preprint or something, because I really want to know the details of how you go about modeling all of that.
The preprint's up, but it's no movement on the publishing side. But the preprint's up. I'll show you.
Really fascinating talk. I'm not an expert of the TF binding, but you mentioned that some TFs can bind to inaccessible chromatins. So that's kind of counterintuitive. I wonder, so if the chromatin is inaccessible, how the TF can access the chromatin and bind to the DNA?
Yep.
That's my first question. And also a relevant question is like, if they bind to the DNA, so whether it's like, how do you validate these TFs are bind to the DNA directly and not bind to chromatin remodeler and then indirectly
very
bind
good
to
question.
DNA? And also, did you look into like say RNA-seq data to explore the functional impact of this kind of binding?
Yeah. Okay, so let me take them each in turn. So first of all, I don't think it's too surprising that we do see factors that bind to sites that were inaccessible. They'll make those sites accessible after binding. But that's how development works, right? Like you have cascades of transcription factors that make cell-specific or lineage-specific enhancers available for other transcription factors. Let's see, the second question was, how can we distinguish that these sites are directly bound by the transcription factor versus tethered through something else? That's a great question. The ChIP-EXO data is quite narrowly specified. It's high resolution, and it's centered for most of these sites, for most of the Fox transcription factor sites, it's centered on a cognate motif. So we're assuming that means there's binding that cognate motif directly. However, there's no guarantees with that, right? We do see other cases with other transcription factors where at that peak for ChIP-EXO, you have a motif for a different transcription factor. So we do think that ChIP-EXO can tell you tethered binding versus direct binding. That's a different story for a different day. And then finally, I think the downstream expression effect is like what I said for Ferhat. We have measured the RNA-seq. You do see differential expression of different genes downstream of each of these Fox factors, and it lines up a little bit with what their natural, developmental lineages are.
Yeah, thanks.
Thank you.
I will squeeze in one last question. It was really beautiful work. I was going to begin by saying I had an absurd suggestion for an experiment, but seeing how hard you've worked at the mechanistic approach, it's not so absurd. So how would you feel about grafting like the DNA domain for FOXP3 into FOXL1? And I presume that the context window for the CNN for the motifs is large enough that it should capture patterns of any possible other TF factors that might cooperate. So the fact that you're not seeing any signal there,
we're not seeing secondary motifs.
Yeah. So then I wonder if that will then demonstrate the sufficiency of the non-binding domain to mediate this interaction.
We are already doing the chimera. Not with other pairs of factors, but like we're swapping in the G1 into the A1. We're doing a couple of things like that.
You're working on that.
We have the lines made already, but just
Good.
not
Okay. All right. So it's not so absurd. Okay. Thank you. And
thank you.
Thank the speaker again.